So the next section, 24.4, is all about stems. So we just learned about roots, so now we're talking about the shoots, part of the shoots, which is the stems. And so at the end of each stem, there is a shoot apical meristem. So think back to the roots. Now at the very bottom of the roots, there was the apical meristem down there. Apical means apex at the end. So the root apical meristem is at the bottom of the root. The shoot apical meristems are at the ends of the stems. And these produce new cells that elongate and increase stem length. So this is, again, making cells divide, like the zone of cell division, and then those cells are elongating, which are increasing the stem length. They're protected by the terminal bud, which is enveloped by this thing called leaf primordia, which you don't really need to know, but they're like a bunch of baby leaves that protect the meristematic tissue because that's very sensitive. That is how plants grow. And if we harm the apical meristems, then they can't, the shoots can't keep growing. Okay, so this is how your book likes to describe it. So here are the terminal buds, and here's another terminal bud because this is a different shoot. And so this is the growth that has happened in 1 year. And you can tell there's a bunch of axillary buds like we talked about before, and there's nodes and internodes. So there used to be old leaves here, but they have fallen off and left leaf scars. So the terminal bud is where the apical meristem is located. So there's 3 types of primary meristems. Sorry, I skipped over this before. There's the protoderm, which will become the epidermis of the shoot tissue. Then there's a ground meristem, which will become the ground tissue. And then there's the procambium, which will become the vascular tissue. So that will become the xylem and the phloem cells. So there's those 3 types of meristematic tissue found inside the terminal bud. So the shoot tip is right here. So this is where the growth and differentiation is happening, and then down here is the more mature and developed tissue. And so here's the 3 primary meristems. On the outside is the protoderm, which is going to become the epidermis, and then on the inside, all this stuff right here is the ground meristem, which will become the ground tissue. So that's where photosynthesis is going to happen, and storage and everything like that. Those are the parenchyma cells. And then these things right here, found in the bundles, are the procambium. That will become the vascular tissue. And so then here's what a mature stem section looks like. So around the outside we have the epidermis that came from the protoderm. Then in the middle we have the pith, and around here the cortex. So that came from the ground meristem. And then these little guys right here are the vascular bundles. So we can see that inside here we have the what will become the xylem, and then on what we have, no, wrong. Oh, your textbook has it backwards. So we'll get there. This picture does not match this picture. So there's a couple different kinds of stems. Well, there's 2, herbaceous and woody. And herbaceous means green. It doesn't ever get tough. So this is kind of like a flower you'd pick out the ground, like a dandelion stem is herbaceous. And they exhibit only what we call primary growth. So primary growth means that it really only grows up. And they have the outermost tissue is their epidermis, and it's covered in a waxy cuticle like we talked about before. So it's this kind of thick waterproof substance that prevents water loss through the outside of the stem. And the stems have distinctive vascular bundles. So you can actually see, going around the inside of the stem, the xylem and the phloem together. And it's different in monocots and dicots. In dicots, the vascular bundles are arranged in a ring, and in monocots, they are arranged just throughout the stem. So this is what a herbaceous dicot stem looks like. So in the middle is the pith. Remember, we did not have a pith in dicot roots, but now we have a pith in a dicot stem. We have the cortex on the outside. So the cortex and the pith are the ground tissue of the stem. Then we have vascular bundles, which you can see around the outside. So here's a vascular bundle, here's a vascular bundle, here's a vascular bundle. And what that is we can zoom in on this vascular bundle right here. On the outside we have the phloem, on the inside we have the xylem. And then all around the outside of the plant, we have the epidermis. And then in here we can see, you can't see it, but it says parenchyma down here. There's the parenchyma cells of the cortex and the parenchyma cells of the pith. And then between here, we have the vascular cambium, which we're going to talk about, but that's what keeps making new xylem and phloem. This is what a monocot stem looks like. So again, it's different. They're not arranged in a ring. They're scattered throughout the entire thing, but they're still associated together. So here's one vascular bundle. You can see the phloem is right here and the xylem is right here. So they're still associated, but they're not arranged in a ring like in the dicot stem. And then we have something else called woody stems. And so this is what you guys would most likely associate with trees. And it is a little bit different because there's extra growth that happens. So they have both what's called primary growth and secondary growth. So primary growth is upward growth and secondary growth is outward growth. And so the outward growth is what takes a little more work. And so there's primary tissues formed each year from primary meristems, so growing upward every year from the primary meristems. And the secondary tissues developed during the first and subsequent years from lateral meristems. So now we're growing outward. And woody stems have no vascular tissue, and so they have three distinct regions. They have what we call bark, wood, and pith. So inside, we have the pith. This is the ground tissue. This is mostly for storage because since it's so far inside the stem, not a lot of photosynthesis is happening there, but it's still ground tissue. It's just being used for storage. Then in the middle we have the wood. Wood is xylem and secondary xylem, and bark is phloem and secondary phloem. So we're going to get into more of what that means. So bark contains cork cambium, and phloem. So these are the outermost cells that have replaced the epidermis, the cork cambium, which is making new cork, and the phloem, because the phloem is on the outside. So if we have the big ring of the stem, the phloem is located outside, the xylem is located on the inside. So the outside, the bark, is cork cambium, and phloem. And bark can be removed from a plant, but it's actually really dangerous because we're taking away the phloem. So we're taking away the means that this plant has to transport sugars up and down. So that's bad if you cut off the bark from a tree because you're taking away its transportation. It's like cutting out your blood vessels. And these cork cells are impregnated with what we call suberin. And suberin is a waxy substance that prevents water loss from happening. And so basically, when you have a woody stem, you're not exchanging water through the stem anymore. That's going to happen in the leaves because we have this tough cork, which is so thickened with suberin that water can't get in and out anymore. So that is no longer the site of gas exchange. Then we have the wood. So the bark's on the outside, and then the wood's in the middle. And the wood is secondary xylem that builds up year after year. So basically what this means is every year, starting at the first year, the plant makes xylem. Now xylem is dead at functional maturity, remember, so it makes new xylem cells and then they die. But they're so thickened with sclerenchyma tissue that they stay there. And so they're tough and they stay there. And then year after year, the plant keeps making new ones and stops using the old ones. But the old ones stay because they're really, really thickened with that lignin. So basically, we just keep making more and more and more, and we're not using the old ones that we used from years before. And so it builds up year after year, and that's what the wood of a tree stem is. And there's a difference between winter xylem and summer xylem because in winter, we're not making new xylem because we're not really doing too much in the winter. And so that's why we have rings inside of a tree, because you're looking at the wood, which is the xylem, and it looks different in the winter than it does in the summer. So you don't need to know that. But these rings right here show the differences. Every year we're making new xylem, and so every year we see a difference between winter and summer xylem. And again, you can see the three different places. So the pith is the inside right here. This is the ground tissue where storage is happening. Then we see right here the wood, which is the xylem and the secondary xylem. So here's the spring wood and the summer wood, and so an annual ring. This is 1 year right here. So the summer wood is different than the spring wood. And so the vascular cambium is found right here. So this is where we're making new xylem and new phloem. So every year this vascular cambia moves outward. So after the first year, it would have been right here. And then after the second year, it would have been right here. After the third year, it would have been right here. Each year we make a new layer of xylem and we push the vascular cambium out one more year. And so that's why you can tell the years on if you cut down a tree stem, because you can see the annual rings. So this vascular cambium right here is making new xylem and it's making new phloem. And the phloem, along with the cork and the cork cambium, are becoming the bark. So those are 3 different layers of a woody stem. And here's a tree trunk. You can actually see the annual rings. So again, that's the difference between the summer and the spring xylem, secondary xylem. Now there's a bunch of different types of stems which you guys can be on your own to figure out. Good luck with stems.